Yep.
And thank you everyone for participating. And Hector, please.
Hey everyone, thank you for participating in our spatial seminar today. Today, our presenter is Dr. John Hickey. Thank you so much for being here. He is an assistant professor at the Biomedical Engineering Department at Duke University, with secondary appointments in the Cell Biology and Biostatistics and Bioinformatics departments of the School of Medicine. His lab is composed of a diverse set of engineers and scientists solving health problems by using and developing systems, biology tools, and technologies in order to describe and control spatial relationships between cells in tissues. John recently completed his postdoctoral training at Gary Nolan's lab at Stanford University, where he used and developed systems biology tools to analyze spatial relations amongst cells and tissues. He has also been recognized by a number of organizations for his work, including receiving the American Cancer Society Postdoctoral Fellowship, the NCI Postdoctoral Fellowship, uh, the ARCS Scholar, a Cybele Scholar, and the NSF Graduate Research Fellowship. Today, he will be talking about his advancements in signal cell multiplexed imaging and its coupling with sophisticated computational algorithms that have really greatly augmented our views into complex tissue microenvironments. And with that, I'm going to go ahead and turn it over to Dr. John Hickey.
Super. Thanks for the kind introduction, and great to be with you guys here on this beautiful sunny day, at least here in North Carolina. But the great thing is we're all virtual together. So I'm going to talk a little bit about how I see tissues, since a lot of us work with that tissues, and kind of the resultant thing that happens from having that vision. So what I'll start with is kind of this, how I see the tissues, which is this multi-scale network of interactions. And what do I mean by this? So cancer could be thought of as a tissue, or any other tissue you take for that matter. There are many different parts of the tissue that are all working in concert in space to perform the function of the tissue. And within each of these different areas of the tissue or organ, there are different cellular neighborhoods, and these are composed of distinct compositions of cells that are also spatially related and in proximity to each other, and in these large networks across the tissue. And then even within that, we have cell-cell interactions between different cells with ligands and soluble factors. And then even within that, at a cellular level, we have intracellular signaling within molecules within cells that control the fate and function of cells. And so it's, network of interactions at different scales of biology, but they're all happening simultaneously. And we've done a good job, I think, of characterizing them individually across scales. But I think, to really understand how to control some of these, especially within the context of these larger scale spatial biology, I think connecting them in how we measure from the ground up is really important. So one example is through Codex multiplex imaging. Here we're able to make 50 molecular measurements at a time. Through quantification, we can identify the different cell types and their proximity to each other. From that, we can do computational analysis to understand the multicellular neighborhoods, and then start to look at network interactions across these scales. So I'll just give a brief intro to Codex multiplex imaging. Basically, it's a technique where you use any number of antibodies that you want to stain your tissue with. To each unique antibody, you give a unique barcode, which is a DNA oligonucleotide. That allows you to go beyond the traditional 3 or 4 and that you can normally do. And the way that you do that is you add a complementary sequence to your antibody with a fluorophore. You image 3 at a time. Then you do a gentle denaturation where you strip off those oligos and add the next set. And then you keep doing that until you've imaged all the antibodies in your panel, which allows you to do 50 plus antibodies at once in tissues. So, needless to say, there was a lot of protocol development, thinking about how you process data, how you standardize metadata, how you develop antibody panels, how you think about different data normalization and how you identify cells correctly, and how do you think about doing that in a, a machine readable way. And we've been involved in a lot of these efforts, but I won't go into those. Needless to say that the data quality that we're able to achieve now is um, because of a lot of these efforts that went into what people might consider maybe boring aspects of technology development. But they enable really cool studies like this one. I'm going to share two vignettes today about using these, codex multiplex imaging to understand how we have tissue organization across scales and how we can think about it in terms of some of the analyses. So one project that I got involved in was we were interested to learn about the human intestine and how different parts of the human intestine were different in terms of their organization and cellular components. And so we took 8 different regions of the healthy human intestine, 4 from the colon and 4 from the small intestine. And we did codex multiplex imaging with a panel of 57 antibodies. So if you haven't looked at some data like this before, here's an example image of the human intestine. Here are markers that just 5 of the 57 markers that we used overlaid. And you can see this purple region is the epithelial area. You have the submucosa underneath it and the muscle layer here. And so what we can do is we have cellular resolution. You can see each one of these is a unique cell. We can look at different marker combinations, right? These are different marker combinations to understand how follicles develop or fade away. We can start to look at intraepithelial lymphocytes. We can look at the crypt area of the intestine and start to stain for many of these different factors at once. And while these are pretty pictures, we want to be able to quantify this in meaningful ways. And so what is usually done is we apply deep learning algorithms to segment out each individual cell and quantify the protein expression at a single cell level. We apply unsupervised clustering to identify the different cell types based on the marker expression. But one of the things that's challenging is that sometimes the segmentation is not perfect, and sometimes there's artifacts in the fluorescent images. And so that confounds this cell type analysis, which requires an expert annotator nearly 20 hours, per sample. And for the number of samples that we were doing in this study, that wasn't something that I necessarily wanted to get into. And so I teamed up with some computer scientists at Stanford, and we developed this algorithm called Stellar, which basically transforms your spatial omics data set into a tissue graph type structure where each cell is a node and has properties of its protein expression and then is connected to its nearest neighboring cells. And using that, we can train a deep learning model to understand patterns both in terms of the protein expression and the neighbors of the cell to be able to predict cell types on additional data sets. And the neat thing that our computer scientists collaborators did, Maria, particularly in Hickey, was develop the algorithm such that even if there was a cell type not present in your original training data, it could separate that out downstream. So this is an example of what the cell type composition from a healthy human intestine looks like on our data set, with each cell colored differently. And if we leave this tissue out and then train on other data sets, we can see that the prediction very closely matches our original ground truth. So we use this to propagate all the cell type labels across all the different regions of the intestine and across all the donors. And there's a lot of things that we can learn in terms of overall cell type composition and how it changes across the intestine. I have a background in immunology, and so, looking at some of the differences in the immune cell, how they change from the small bowel to the colon was interesting to me. And particularly, you can see here that the CD8 cells are changing. But one of the things that was also interesting to me was that even though there were higher numbers of CD8s in the small intestine compared to the colon, there was a high degree of variance between donors in terms of the percentage of CD8s within the small bowel. And when we looked at our metadata, we interestingly found people who had a history of hypertension had a lower abundance of CD8 T cells within the small intestine. And this was a little surprising. So we also checked endothelial cells, which would make sense that there would be a lower number of endothelial cells in the small intestine. And indeed, there was with a history of hypertension. And, specifically, we looked into the spatial area of where these CD8 T cells were particularly lost, and that was in the intraepithelial lymphocytes. And so, these are implicated in a number of diseases. And so it would be interesting to understand why these CD8 T cells are being lost, and B, what effects that has on overall biology of the small intestine. Another thing that we noticed with the metadata that we had from these healthy donors was that the percentage of M1 macrophages within the donor had a strong correlation with the body mass index. And what's really interesting of this is body mass index is known to be associated with gastrointestinal diseases, and perhaps M1 macrophages as an inflammatory macrophage phenotype is an early indicator of potential gastrointestinal disease, because none of our donors had a history of gastrointestinal disease, but this could indicate a role. And we looked at spatially where these cells were, and the MT macrophage counterpart was distributed across the different areas of the intestine, whereas the M1 macrophages was primarily restricted to the mucosa. And this prompted us to start to work with Magda, who's also at Stanford, and start to look at other flavors of macrophages. She was able to characterize several different flavors in terms of more than just M1, M2 split from single-cell RNA-seq data, and then characterize where those specific macrophages were within the colon, breast, colon cancer, and breast cancer samples. And we again established that distinct flavors of macrophages have distinct microenvironments. So I think a lot to follow up there as well. So, a lot of what I've told you so far is, if we're thinking about recreating something complex like this cool Lego set, mostly what I've told you so far is just categorizing things into different pieces and quantifying the pieces. But, the spatial data really presents an opportunity to look at how cells fit together, right? And how they interact with each other. But beyond that, I think what is extremely important is not only cell-cell interactions, but potential multi-cell modules and how these come together to form functional tissue units. And so with that in mind, the lab has developed a cellular neighborhood algorithm where you can take these windows of cells, look around a window of cell, and turn this into a vector, basically the composition of what cells are within this window. And when you do this, you can then cluster those vectors in a similar way that you cluster cell types and start to identify similarly composed different regions of the tissue, which we call cellular neighborhoods. So what does this look like on tissues? Here's the fluorescent data from the human intestine. Here's the cell type map. And then if we do our cellular neighborhood analysis, we can start to see, we can identify things like different follicle regions, the vasculature, as shown in yellow, and different epithelial areas. And what does this look like quantitatively? We can start to look at these neighborhoods where different cell types are enriched or depleted. For example, this plasma cell enriched neighborhood is enriched in plasma cells, CD4 cells, and some antigen presenting cells. And, we can propagate this to all of our tissues and start to compare how the different neighborhoods change across different conditions. But, the thing that's interesting to me to think about is, even beyond these cellular neighborhoods is, you can think about the vasculature that's here versus here next to the follicle versus here in the epithelial area or the mucosal area. And these vasculature are all very different and in different contexts. So how do we think about analyzing different hierarchies or hierarchical spatial domains? And, in geography, we already have this, right? So we have different neighborhoods that can make up a county, and then we can zoom out and look at different counties that then make up a state, and then different states that make up a country. And so, I think it's important when we think about this spatial analysis to think about these different levels of geography for our cells as well. And so that's what we did for this intestinal data set. Here we have an example of a small intestine section, and here a colon section. And you can see that here we are characterizing the cell types are the colors. Then when we do neighborhoods, we can start to see how the neighborhoods are distributed across the tissue, but then we can look at neighborhoods of neighborhoods, or what we call communities, and then at a very high level cell, keep going to define tissue units. And this is all from the ground up. And so because we're doing that, we can connect basically what cell types are enriched in which neighborhoods are enriched in which communities, and which communities are enriched in which tissue units. And so this can help give you a very quick and informational overview of the structure of a tissue across different scales. We can learn a lot from this, but what I will just focus on is one example where this adaptive immune enriched community is the intersection of multiple epithelial and immune neighborhoods. And one of the reasons that we thought it was particularly interesting was that its placement in the tissue. So you can see it's this orange neighborhood, and it's found at the base of the mucosa. And this is an area where it's the stem cell niche of the intestine. And what we found was that there's an enrichment for adaptive immune cells like CD4 and CD8 cells towards the bottom of this crypt of the mucosa. And that wasn't something that was previously noted before. But the other thing that we noticed, which is really interesting, is that for the communities, they're layered as you go up, right? You can see this layering as you move closer towards the lumen of the intestine. And it's known that epithelial cell populations change as you move closer to the lumen of the intestine. But because we are able to quantify these into different zones, we also were able to understand that the immune cells, so these are all the different immune cell types, these colors, they also change as you move closer to the lumen. So not only is the intestinal mucosa structured in terms of how the epithelial cells develop as they move closer to the lumen, but also in terms of the immune microenvironments. So another student in the lab started to work alongside another PhD student who was in statistics, and thought about, how can we use single-cell RNA-seq and single-cell ATAC-seq that we have from the same donors in a more synergistic way rather than, just an additive way where we look at things separately. And so they developed MaxFuse, which is an algorithm for basically matching data sets that have weakly shared features, meaning they have shared features, but not all features are shared, and they're not the exact same modality. So in our case, we have single-cell RNA-seq and also codex. And so what it basically does is it learns this association by using all of the data set, so understanding the local neighborhoods within individual components, and then iteratively matching those smoothed modalities between each other and propagating those across the different cell types. So what does this look like? So we have the small intestine and the colon for single-cell RNA-seq in this co-embedded space. We also have the codex multiplex imaging. And if we look at the cell type, we can see that there's good agreement in terms of the overall cell type within both assays. And then we can also look at, for just one cell type, for instance, the goblet cell, where we can look at the protein and RNA expression. So for example, goblet cells should express MUC2, and we see that both within the RNA and also in the protein space for both the small intestine and colon. And so now with the data integrated at the single cell level, where each individual codex cell has associated single cell RNA and ATAC profile, we thought, what can we do with this integrated data? And, we've already computed these cellular neighborhoods. And so what we did was we could extract these different cellular neighborhoods and compare the RNA expression for these different neighborhoods. And, you generate something like this, which is an eyeful, but just, which is a number of genes that are differentially expressed within each individual neighborhood as compared to all others. So if we take one example like the outer follicle and put these differentially expressed genes into a gene ontology enrichment, we can start to see a lot of different processes we would expect within a follicle-based neighborhood. So now we're able to differentiate the neighborhood-related gene profiles in this way. So just to conclude this portion of the talk, we've this multi-scale nature of the tissues and how we calculate that and compute it and describe tissues, I think is really important. Another thing, thinking about different cell types being spatially restricted and having different phenotypes because of their spatial restriction, I think we'll continue to discover more of these. And then, I think it's interesting that we see immune structures and regular immune organization just as much as we do within the epithelial space of the intestine. And then more of this integration of multimodalities will help synergize in understanding complex tissues rather than an additive approach. So for this next portion of the talk, I wanted to get into more of an application. That was more understanding of tissues that were deconstructing the complexity of nature. Whereas here, one of the things that I wanted to learn is how do we apply perturbation to a tissue microenvironment and start to understand what's changing versus not. And a perfect setting for this is in cell therapies. And, particularly I'm interested in cell therapies because I think that there's a lot of really interesting things we can do with them across a number of different disease conditions. They're programmable, you can quantify them, you can control them to some extent. They're living, they can have memory, they can interact with other cells, they can traffic to different places, they can have multiple payloads. And so there's a lot of really amazing benefits with cell therapies, but these benefits of cell therapies are also a lot of the challenges with cell therapies right now. They can traffic to unwanted places, they can take unwanted inputs and change their function, they can have off-target effects. And so there's a number of challenges with cell therapies, and a lot of these are related to their spatial interactions. There's a good market for cancer specifically for cell therapies. And it's not just CAR Ts, and it's not just Ts, there's lots of other different types of cell therapies that are coming out. And it's not just specific B cell targets, there's a number of tissue-associated antigen targets as well that are being moved forward. So just to set up the experiment that I did and quantified was there's lots of different flavors of T-cell therapies, but we used an engineered TCR, which basically is a TCR that recognizes a specific antigen. And in this case, we're looking at the melanoma antigen GB100. And so what we do is we take out these antigen-specific T cells, we grow them in a dish, we expand them to high numbers, and then we throw them into mice who already have a melanoma growing tumor. And then we can, let that therapy work for a number of days and then harvest the tumors for codex multiplex imaging. And so, we did this and, we wanted to understand how does a T cell therapy impact the tumor microenvironment? And so we took out the tumor at different days. You can see some of the progressive inflammation as highlighted with the red and yellow colors, which are MHC1 and PDL1, both indicative of increasing inflammation, and also some of these other immune cell markers. But not only that, we can quantify this over time and start to understand how does cell therapy expand and how does it potentially the tumor overcome that response. But, the thing that I was really interested in was, we can understand these dynamics, but what's happening to the cells specifically that we're throwing in, which are these CD8 T cells, which are having the effect of the tumor microenvironment. And so, thinking about how do we quantify the spatial features of a cell, I thought, in the T cell field, it's very common to profile cells by their molecular features and then quantify them into different phenotypes like memory versus effector. And so, I thought, why not do this with spatial features? And so essentially that's what I used to cluster them as opposed to molecular features. And with that, we can create a spatial UMAP, for instance, where each individual data point is a distinct T cell within the tumor microenvironment. And the relationship from one point or one T cell to another represents how similar the microenvironments are to each other. And so, data points here and here might be very similar in microenvironment versus a data point over here, which is a very different data microenvironment. And we can look at this, from day 0, 1, 3, 5, and 12 to see how cells change in their microenvironment over time. And we can cluster these microenvironments, similar how we cluster cell phenotypes, to understand what are the major compositions or microenvironments that T cells are seeing in their time in the tumor. And then because we've clustered these, we can then quantify these neighborhoods over time, and start to understand what are these natural progressions of the T cells where they infiltrate, they associate with a high number of immune cells, particularly dendritic cells. They migrate further into the tumor to cause this tumor inflammation and killing. But then, the tumor eventually starts to outgrow this, at least in this experimental context. And so, because the tumor in this context was outgrowing the T cell therapy, so the T cells were having some effect, but then the tumor was outgrowing, we thought, well, what can we do to change that? And one of the things we looked at first was, okay, what are the cells that we're looking at, endogenous versus transferred? And so we stained them with this marker that marks cells that were transferred in. We can see that there are both transferred T cells within the tumor and also endogenous T cells within the tumor. And we quantified this. This is nearly, most of the cells in the tumor are these ones that we've transferred, but there is a good percentage of those that are not. And, interestingly, if we look at cells that are PD-1 positive, most of these are cells that are endogenous. The cells that we transferred in were less PD-1 positive. And so we looked at other things that could be correlating here. And interestingly, we see a high percentage of our cells that are CD25 positive, whereas we did not see that for our endogenous cells. So there's a number of markers that potentially we can use in the future to separate out these transferred or antigen-specific cells from the endogenous cells. But not only that, we saw within different percentages within different neighborhoods where the transferred cells were most within these inflamed tumor-enriched neighborhoods. So areas where the T cells are going and actually killing the tumor cells and not just in these immune-enriched neighborhoods. And so, that didn't specifically suggest anything engineering-wise that we could do to the T cells, but there's a lot of people, including my kids, who love superheroes, and making super T cells is a very hot topic, and there's many ways to do this. But one of the ways that we came across was another group had inhibited acetyl-CoA production, and saw that there was a distinct difference in terms of tumor growth, where when you inhibited this acetyl-CoA in the T cells, then you got much better killing. And so we also applied this in our hands, and we also characterized the phenotype after we applied this treatment, and we get much higher memory T cells compared to when we don't use this drug to inhibit acetyl-CoA. And so, we did the Codex multiplex imaging, we did all the quantifications, and using a random forest model to kind of separate out what could be the most important things or cell types in predicting whether you have a better therapy or not. Maybe not so interestingly, or is that some of the tumor cell types came out as most important, and CD8 cells. But the most interesting thing to me was when we take these percentages and we look at the percentage of proliferating tumor cells versus these inflamed tumor cells, we see that there's a negative correlation across all samples, irregardless of the type of treatment they got. And to me, this indicates that there's a phenotype change from a proliferating state to an inflamed state. And then when we look at it, the inflamed tumor cell population compared to CD8 cells, there's a positive correlation indicating potentially CD8s are playing a role in converting proliferating tumor cells to inflamed tumor cells. So, I mentioned this important multi-scale network of interactions and how I've been showing how we've been deconstructing it through looking at it through the lens of codex multiplex imaging. But I would argue that it's just as important to reconstruct the complexity. And you can do this in terms of in vitro models, but also in terms of in silico models. And here, what we did is we built a multi-scale agent-based model. And essentially what you do is you define a cell as a computational object. You give it rules and properties. You allow it to interact with other cells within a spatial microenvironment. And then you allow the dynamics in a simulation to have this large tissue behavior emerge from it. So we built such a multi-scale agent-based simulation, and particularly we were motivated because the codex data provides such great input to such a model, right? We know the XY positions, we know the phenotype of the cells, we know the cell types, and then we can use that input to guide an accurate simulation of what the dynamics might be, because codex only takes a snapshot of these tissues. And so we can start to simulate, a tumor growth without any T cells. We can simulate when we transfer T cells, how do they kill? Where do they kill? How do they migrate to the tumor? And if we change the phenotype of the T cells, we can start to understand how they're controlling tumor better and creating these different tumor neighborhoods. And then additionally, we can quantify this dynamically over time. One of the things that we see if we change the phenotype, just like what we did in vivo, is that we have a much greater control of the tumor. But surprisingly, it wasn't necessarily because the T cells were killing any better. There's this large discrepancy in terms of tumor number versus the number of tumors that T cells were killing. And so, the videos were playing really fast, but the T cells, these little green dots, there's a high percentage of them next to these tumor cells that have been converted to an inflamed tumor type. And so essentially what was happening is that the T cells in this changed phenotype were able to convert more of the proliferating tumors at an earlier time point to this inflamed phenotype, allowing the T cells to catch up with the exponential growth of the tumor. And this was more important than necessarily having increased killing ability. And, so I think that this theory experiment loop with models and imaging is complementary in terms of adding dynamics and adding important, initial conditions for modeling, and helped us to understand that really this tumor phenotype control is absolutely essential for designing cell therapies and also having effective ones down the road. And I didn't get into it today, but also how they preserve their phenotype by controlling their microenvironment. So the other thing that we noticed that was different between these metabolically treated T cells versus just the normally active T cells was that the microenvironments that the T cells are found were different. And one of those was this where they contact the tumor or where they're killing the tumor. And in the regular T cell activation, we see an enrichment of Tregs and a loss of MHC1 on tumor cells, whereas we don't see that in the 2HC T cells. And what could be, I talked about molecular versus cellular, and what could be some molecular features driving this. We looked at RNA-seq of these T cells, and we see, things that we might expect in terms of the phenotype that differentiates these two cell types. But interestingly, there's a number of things that drive migration of other immune cells and also their own migration that separated these cells. But what was most interesting to me was that there's a difference in terms of their cytokines they secreted. And particularly, we see the regularly activated T cells secrete a lot of anti-inflammatory cytokines like IL-10. And we profiled this in CyTOF, and it's not all of the cells. It's only a fraction of the cells, but this fraction of the cells was enough to cause some of these anti-inflammatory neighborhoods within, where they're attacking the tumor. And, this is another T cell spatial UMAP. And essentially what we see from effective T cells is they're moving from the vasculature enriched in these immune infiltrate neighborhoods that are proximal to the inflamed tumor. And in data that I'm not showing, it's the size of these neighborhoods that are in proximity to the inflamed tumor that allows the T cells to have these productive environments deep within the tumor. So I will just skip some of this since I want to save some questions or some time for questions. But essentially, we also looked at this within metastatic melanoma patients who received checkpoint blockade therapy, and 3 who responded versus 3 who didn't. And we had baseline and follow-up, and we're able to compare the spatial responses within these. And the short story is, and I'll skip some of this, is that we see the same cellular organization within responders as we did within the effective T cell treatments within the cell therapy within mice. And specifically, we see at these zones where the T cells are causing tumor death and also causing tumors to become inflamed, they're found next to a dense layer of this immune infiltrating neighborhoods. And you can see some of that here where the tumor cells are inflamed and you have a lot of T cells, and they're next to this very dense immune infiltrating area. And we also quantified this through permutation testing, and see this only within responders and not non-responders. And the size of these immune infiltrating neighborhoods was much higher in people who or post versus pre-therapy as well. So the checkpoint blockade is having a increased ability to enlarge these cellular neighborhoods, which are, distinct compositions of immune cells over large areas of the tissue. So in general, this portion of the talk, we used multiplex imaging to really characterize a lot of dynamic responses in cell therapy and patient responses in melanoma. And, we saw that this T cell phenotype conversion was really important for overall survival or efficacy, but not only that tumor conversion and killing that was happening in this neighborhood, but also that was supported by a large immune infiltrating cellular neighborhood, distinct from other immune cells besides CD8 T cells. And T cell therapies that did not do well or immunotherapies that did not do well had this neighborhood, the same neighborhoods, but they were more fragmented and less contiguous. So, I mentioned the intestine, I mentioned the cellular therapies, but we're seeing, other instances such as in Barrett's esophagus, where immune cell organization is controlling or contributing to disease progression or overall tissue structure. And so just to conclude, I think that infusing time back into the static data sets, ways we can do that, like with the multi-scale modeling, is going to be important for us to gain insights. Thinking about our cell therapies from an anti-proliferative as well as a killing program will be important in our design in the future. And then the ability to change a local and microenvironment, is going to be another design consideration. And then, thinking about cell therapies, even a fraction could have a detrimental effect if they cause disruptive neighborhoods within the tumor microenvironment. Like to thank a lot of people. Most of the work happened across many different labs and with a collaborative effort with many different people. Like was mentioned at first, I just started my own group at Duke in the biomedical engineering department, and we do a lot more of this type of work, including engineering of cells and materials within the context of spatial biology and computational development. And with that, I'll turn it back over for questions.
Thank you, John, for the wonderful presentation. So, are there any questions?
So let me start. There's a lot that you presented, a lot to unpack. So let's start with the first part of it. Did you see any differences in different parts of the colon or different parts of the small intestine? Because as you know, it's a 10-foot long organ, the small intestine. The different parts are presumed to have different roles. So were the immune compositions of these different parts different? Do we have any insights as to why tertiary lymphoid structures form at certain areas and not at other areas? What were the vascular differences? There's a lot that I can ask, but I'll stop there for the first part of the presentation.
Yeah. So great question. And there were differences, and there are differences, particularly within the small intestine. There are greater variation than within the different colon segments. And in terms of compositions, there was many. And we can get into what those were in different the percentages of cells, at least, were very different between those. Now, in terms of the composition of those cells and the organization of those cells, that also changes slightly as you move from one area to the other. And we only looked at a very small fraction of those interactions. And so I think that the data is rich to explore even more of, those minute changes across the different segments of the intestine. But in general, we saw differences mostly within the different regions of the small intestine.
And then just to continue, and again, I don't want to monopolize, but just to continue on the second part, did you see in humans, when we treat disease, you get differential responses in different metastatic sites. So did you have an occasion in your animal models to see differential response to the T-cell therapy that you have, and any insights that you were able to draw, or are there plans to do something like that?
So I'll just repeat the question just to make sure I understand. Are you saying that in the animal model, did we see differential responses in different tumors? And then could we correlate that to any changes in the cell microenvironment? So yes, I would say the changes that we saw in terms of efficacy were due mostly to being able to have more T cells infiltrate these tumor zones and then cause an inflammation and killing within those tumor zones, but have large areas of that, conserved areas across the tissue, and then have next to those, or abutted next to those areas, highly immune infiltrated zones. So there's a high number of CD4s, dendritic cells, macrophages, and T cells as well. And that was one of the main differences that we saw. We didn't in the animal model, just because we we didn't take samples of the tumor. We had to sacrifice the animals and take the tumor. We didn't like separate people into like responder or non-responder, but we just had therapies we knew would work or not work better.
Sure. Thanks.
Yeah.
Wonderful talk.
Yeah.
Other
Thank you.
questions?
Are there any other questions? Please feel free to unmute yourself or ask.
Oh, can I ask over here?
Yes, please, Arthur.
Yeah, thanks so much. I was actually going to ask about this spatial UMAP, specifically the one that you run it on a T cell neighborhood. Is it really the UMAP that you learn on a spatial graph, or you just call it a spatial UMAP, or is it just just kind of a two-dimensional embedding that captures the neighborhood relationship?
Yeah, good question. So it's basically the embedding of the spatial relationships or the spatial features of each individual cell. So it's not necessarily spatial in the sense of like it's re-representing the tumor positioning of the cells, if that's what you're asking.
Oh yeah. But you then you run regular UMAP on it, right? Once you have the neighborhood relationship, right? Or you
Yeah,
just call it
so basically, yeah, we pull out the vectors basically of the neighbors around each individual T cell, and then we use that as input to a UMAP type of algorithm. Yeah.
And the clustering is not on the UMAP, but it's on this vector space that you generate.
Correct. We process it with like, so an example could be, you can use any type of clustering, but in this case we were using Leiden clustering.
Thanks so much.
Yeah.
Wonderful talk.
Yeah.
Thank you. Any other questions? Maybe as we wait, I'll ask a quick question, John. For the multi-scale modeling part, it's a wonderful idea. So have you considered benchmarking it in real, like in real models, or how does that work? I'm curious how they will agree with the physiological systems.
Yeah, good question. I think that it's really hard to benchmark in terms of like a dynamic sense, just because we don't have dynamic, imaging tools, except for, there's some intravital microscopy data that we can get. And so we tuned a lot of our model parameters based on intravital microscopy type data. We did validate it in the sense of, we had that time scale of different tumor neighborhoods that we took out at different time points. And we see similar numbers and percentages of cells at those different time points. And then the other thing that we see similar are the similar arrangements of the cells, because we did neighborhood analysis on these same simulations, and we see similar arrangements. That being said, the models that we have started to build are very simple. They don't have all the cell types within the, tumor microenvironment that we know about. And so, as we get more complex, I think, maybe the comparisons will get more tricky, or maybe easier to compare and say, this is what we expect. But yeah, great question.
I think that's a wonderful direction to restructure the data again, right? From what you learn.
Yes.
Thank you. Any other questions?
So again, since there's no one asking questions, maybe I can push you a little bit more. Any idea about immune infiltration and tissue heterogeneity as to, even in the normal tissue, I'm just trying to come back in a slightly different way. So you said that towards the base of the crypts, there's where the stem cells might be, you are seeing a particular pattern. Is this uniform, or is it related to anything, or any thoughts as to what's going on?
Yeah, I think so two questions, I think. First, I'll answer the second one, which the adaptive immune cells, that's a universal thing that we see across donors. And so there's a number of things that we see in terms of structures that are universal across different donors. But that being said, we also see, slightly different compositions within these distinct zones, if that makes sense.
Yeah.
And an example is, those M1 macrophages, they pop up within the top part of the mucosa for certain donors, and they don't so much in other donors. And the heterogeneity, I think, is a great question because maybe we're losing some really interesting biology by lumping everything together and clustering everything together. And, an interesting way to do that might be by comparing neighborhoods clustered by donor. It would take a lot more time, but, I think that could show, some of this heterogeneity within major tissue structures, right? Like I think that the overall like core composition of these structures are fairly similar to one another, but the heterogeneity is definitely a thing both within healthy and especially within cancer tissue.
There's a question in the chat, Amit.
Oh, chat. Okay. Eliza, would you like to unmute or should we help? I think we can help. I'm not sure whether I missed it or not, but do you believe something like velocity analysis and trajectory inference as done in single-cell RNA-seq would be helpful on your T-cell spatial UMAP? I'm asking this to understand how we can possibly go to spatial temporal analysis.
Yeah, I think that ideas like that can really be interesting. I think it's important to think about, as we're developing such algorithms for spatial, to think about ground truths that you can, be satisfied that whatever, velocity analysis or, pseudo time you're creating, that it actually recapitulates true biological processes that are happening. But yeah, I definitely think that's an interesting avenue to continue.
Wonderful suggestion. Any other questions? I think in the interest of time, let's thank Dr. Hickey for this wonderful presentation. In the virtual audience, lapses are, more digital and all that. Thank you for participating today. And just please know that the seminar series is bi-weekly. In 2 weeks, we'll announce another rising star in specialomics for the speaker, right? And then please watch out and check our social media, and we'll be keeping a note of this. And this seminar was also organized by Sunbelt, which is technically, anyway, we got sun, right? We'd like to highlight specialomics, and also it's a good symbol for the specialomics rising. And also Dr. Badwe from Emory Winship Cancer, Dr. Gukman Polar also Winship Cancer are co-organizing, and also Hector Torres are the program coordinator. And thank you, everyone, and hope to see you in 2 weeks.
Thanks, Amit.